this video, we'll be looking at the regulation of blood glucose level. So what we're going to be looking at is seeing how the actions of alpha and beta cells and the hormones that they release, and seeing the actions of these hormones on different target cells. But before we do that, there are three new keywords that will appear in this chapter and that you need to know what they mean. So first of all, let's go through these three keywords. Now, the first one is called glycogenesis. When looking at these three keywords, it's quite a nice way to do this is by splitting the word up at different parts and then looking at what the different parts actually mean. So, for example, in the glycogenesis, you can split them up into glyco and then genesis. Right, so the way to think about it is glyco usually means glycogen or refers to glycogen. So this is something to do with glycogen. Genesis, like the first book in the Bible, so Genesis means the creation or making of something, right? So the word glycogenesis, the word actually means the making of glycogen, and that is usually made from glucose. So glycogenesis means converting glucose to glycogen, making glycogen. Now the second word is slightly longer, it's called gluconeogenesis. Now again, we're going to split the word up. So we got gluco, then neo, then genesis. So the prefix gluco usually refers to glucose. I mean, it's kind of similar, so hopefully it's obvious. Genesis again means making, so we know that already. Neo usually means new or something different. So gluconeogenesis means that you're making glucose from something different or something new. So gluconeogenesis, to give it a definition, is the conversion of amino acids and glycerol or proteins and lipids to glucose. You're making glucose from something slightly different. Now the final word is glycogenolysis. Now it sounds weird, but I mean, hopefully it's a little bit more obvious as well. So first of all, we already got the word glycogen in it, so we know what that means. Now lysis means breaking. So glycogenolysis means converting glycogen to glucose or breaking glycogen up to make glucose. So here are the three words. Just to recap, glycogenesis means converting glucose to glycogen because you're making glycogen. Gluconeogenesis means that you are making glucose from other materials like amino acids and glycerol. Glycogenolysis means that you're breaking the glycogen up to make glucose. So as you can see, for all of these three reactions, it has something to do with the level of glucose that we can find in the bloodstream. So for example, in glycogenesis, it's about reducing the blood glucose level and you're converting them into glycogen to store them, whereas gluconeogenesis and glycogenolysis is about increasing the level of glucose in the bloodstream. So in the case of the action of insulin and glucagon, we can relate that. We can talk about how these two hormones would lead to these three different types of actions. So now let's have a look at actually how do the different cells and the hormones regulate blood glucose level. So to start with, we will have a normal blood glucose level, now roughly at about 90 milligrams per centimeters cube of blood. And obviously different things that we do in our daily lives, it would cause our blood glucose level to fluctuate. We will never keep it at the same 90 milligrams per centimeters cube level. So for example, after any meals or anything that you eat, then obviously that will cause your blood glucose levels to rise. And sometimes, and after that, usually your body will then regulate that to bring it back down in order to help your body use and absorb those glucose to actually use them well to release energy, for example. And sometimes, let's say, if you're exercising intensely, then your cells are using up that glucose a lot quicker than normal, then your blood glucose level will drop, or maybe after a really long time of not eating anything like fasting, then your blood glucose level will also drop. Then your body will try to do different things to bring that back up because it's important that you maintain that water potential in between the blood and the cells and making sure your cells have enough glucose to function. So we'll look and see what would happen if the blood glucose levels goes up and how do we bring it back down, and if it goes down, how do we bring that back up. So first of all, let's talk about it bringing up. So if the blood glucose level rises, the beta cells would be the ones that detect this. the beta cells they have a glucose transporters on their cell surface. So if there's a lot more glucose than normal, then those transporters will be able to move those glucose into the beta cells and then causes the release of insulin. So I will cover that in another video. Now after that, the insulin then travel to target cells. So specifically, they will target the liver and most body cells. Then they can cause a variety of different things. There are 5 specific things that you will need to know here. Now, the first thing that they do is that they can decrease the level of glucagon released by the alpha cells. Glucagon is the other hormone that I'll talk about in a bit, and that's the hormone that actually increases the blood glucose level. Now, if we're trying to decrease it, obviously we want to stop them from acting on these cells to increase the blood glucose level, so therefore we would stop the release of that, or at least dampen it down. Insulin can also cause an increase in glucose uptake into the cells. So the cells are removing the glucose from the bloodstream a lot quicker, a lot more than usual. And because there are more glucose in the cells, and obviously more glucose can be used up or reacted in respiration to release more energy. So that's also why sometimes for some people with a high blood glucose level, they become a little bit more hyper than usual. It's because they have more energy being released. There is also an increased rate of glycogenesis. As I covered earlier, glycogenesis is the making of glycogen, so you're converting the glucose into glycogen once they're inside a cell. So rather than using them up in respiration, you can store any excess glucose into glycogen, so it's an energy store. And then you use them when you actually need them. You can also increase the conversion of glucose to fat, or at least the rate of conversion of glucose to fat. So you're making more fat instead of glycogen. But in the same case, fat or lipids are also a good energy store for the body. There is actually a name for this called lipogenesis, but it's not covered in the specification. But remember, lipo, lipid, and genesis, creating. So lipogenesis means you're creating fat from the glucose. So these are the 5 things that insulin can do, or the effects of insulin can have on liver and body cells. And by doing all of that, they are reducing the level of glucose in the bloodstream, therefore you would lower the blood glucose level. Now just to summarize this part, now we normally have our blood glucose level at a normal level, but if there is an increase in our blood glucose level, then beta cells will release insulin that will travel through the bloodstream to get to liver and body cells, and they can do these 5 different actions to reduce our blood glucose level back to normal. So this would happen, let's say, after a meal or something that you have eaten. Now obviously then we have to talk about the other end, is when we have a decrease in blood glucose level. When your blood glucose level drops after, let's say, you're doing a lot of exercise or fasting for a while, then your alpha cells will be able to detect that, and they would release glucagon instead, which is a different hormone. And this time the glucagon will target liver and fat cells, because these are the 2 main types of cells that would have a higher store of glycogen and lipids in them. So the idea is that glucagon can affect these cells to cause them to convert all of those storage, like glycogen and lipids, back to glucose and release them into the bloodstream. So there are 3 main things that they can do. First of all is an increased rate of glycogenolysis, so more of the glycogen is broken down at any one point. The second thing is to decrease the intake of glucose into cells, so more of the glucose stays in the bloodstream to maintain that water potential. And that's some of a weird concept, because we often think that, we should have glucose in our cells. But the thing is, if there is an imbalance of that one, then it will cause a change in the water potential in the bloodstream compared to the cells. So for example, if there is, in the case of a high blood glucose level, there's too much glucose in the bloodstream compared to the cells, so the cells would lose the water and they would go into the bloodstream, so your cells become dehydrated. Now obviously in the other case, if there is too little glucose in the actual bloodstream, and therefore again a shift in the water potential, there's a higher water potential in the blood, then the water will move into cells, and if it's too much, they might burst some of the cells. So it needs to have a particular balance to it. And the third thing is an increased rate of gluconeogenesis. So it's about making glucose from glycerol and amino acids, so from proteins and fats. And all of these would increase the level of glucose in the bloodstream and therefore bring it back up. So this diagram summarizes how our blood glucose level could be regulated if there's an increase or decrease. One more thing that is worth mentioning is that the release of insulin and glucagon are both controlled by negative feedback. So in that sense is that the beta cells and alpha cells can both detect the level of glucose in the bloodstream. So in the case of a high glucose level, they release insulin and do all of this stuff to bring it back down. The beta cells would detect the reduced level of glucose in the bloodstream and to stop the release of insulin. So if we look at the control of the release of insulin, then you can see how that might happen. Whereas it's the same for alpha cells, so they release the glucagon to increase the glucose level, and when it gets to the normal level, they would be able to detect that and stop the release of glucagon. So this is regulated by negative feedback to ensure that we won't overshoot in both cases. And this is the regulation of blood glucose level.